Hello everyone, and welcome to this audio podcast. This audio podcast is part of the conglomerate. This audio podcast is part of a Lobostic Shystorm. This audio podcast is part of a Storm and Battle and Chronicle. This audio podcast is part of a Stormish series or edition. This audio podcast as part of a Storm Lobostics production. So, listeners and viewers, the audio podcast today was to be an audio podcast sort of earlier. This audio podcast is will look at is in around and will look at a condition in around where 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 three different organs can be affected can be affected owing to the sort of owing to the sort of how certain pathophysiology affects particular cells in primarily around renal pathophysiology, but then additionally in around the eyes, in around the eye organs, and also in around the ears as well. So today this audio podcast will look at Alports syndrome. So and as part of this audio podcast, there will be a look at an intro to Alports, a look at what are the sort of genetic types of Alports syndrome, a look at X-linked Alports syndrome, a look at autosomal forms of Alports, the signs of symptoms of Alports, diagnosis of Alports, diagnosis and recommendations, and then a look at sort of renal biopsies, an overview of renal biopsies, Alport syndrome, Alport syndrome, sort of pharmacotherapeutics, and then not, and then a novel pharmacotherapeutics, and then in this, and then through this audio podcast, there will be a summary. So to begin with some intro, Alports is generally or has been evidence in the literature as sort of a somewhat rare genetic disorder or condition of specialized basement membrane in amongst the kidneys, the ears, and the eyes. It is a hereditary disease or condition of the glomerular basement membrane, characterized by the familial occurrence of progressive hematuria and nephropathy. About up to 2.3% of all recipients reach ESRD or ESKD, associated with the mutations in one of the genes in one of the genes CO4A3 and COLA4A4 on chromosome 2, or COL4A5 on chromosome X, is and have been pertinent sort of analysis for sort of the genetics of some of the genetic basis of Alports, and then macroscopic hematuria is sort of a major sort of finding of the condition. Other sort of symptomologies include where Alports and can occur with increasing age, where there's renal insufficiency, and there's in around the presence and severity of external symptoms such as deafness and ocular changes. The disease, or the condition of Alports sort of damages the tiny blood vessels in the microvascular in amongst the renal pathophysiology. Alports also causes damage in renal pathophysiology by attacking the glomeruli, and mutations cause the changes in collagen. Collagen is responsible for normal structure and function of the kidneys, eyes, and ears. In as part of the Alports and genetics. So there are three main aspects in around Alports genetics. There is X-linked Alports syndrome, or XLAS. There is secondly autosomal recessive Alport syndrome, or ARAS, and then there's autosomal dominant Alport syndrome, or ADAS. In around X-linked Alports, the accounts or literature has informed it accounts for about 85% of Alports cases. In males, the Alports is severe. In amongst pediatrics or children or young people, there are symptomology that increase steadily with age, and observed and where the symptomologies are similar to that of nephrotic syndrome, and constant, and where there's constant progression into ESRD or ESKD. Asymptomatic paramacular yellow dots and flecks are specific markers of Alports. Analysis of type 4 collagen chain expression in kidney skin and basement membranes using have been pivotal, using or utilizing monoclonal antibodies, and without pharmacotherapeutics or specific pharmacotherapeutics. 90% of males develop renal insufficiency by 40 years of age. Females develop renal insufficiency less frequently, but more slowly. In autosomal Alports, there is associated with homozygous or compound heterozygous mutations in COL4A3 or COL4A4. The disease or the condition is severe in females as in males. Early progression toward there's early progression toward ESRD or ESKD, and there is hearing defect, or can be hearing defect, and retinopathy. Some recipients are free from symptoms or symptomologies, whereas many others display intermittent or permanent microhematria or permanent microhematria associated within glomerular basement membrane. Autosomal recessive Alports is where both parents carry the abnormal genetics or genetics, and both parents pass the abnormal gene to the child. Autosomal dominant Alports is where one parent has the condition, and that parent passes the abnormal gene to the child, and only one copy of the mutated gene is required to cause Alports. In around Alports Alports and signs and symptoms. So with all types of Alports, in all types of Alports, renal pathophysiology is affected. The blood vessels in the kidneys, in glomeruli of the kidneys are damaged and cannot filter the waste, and sort of and sort of any extra sort of toxins from the blood. The common signs of the disease or the condition are hearing issues and abnormalities with the eyes. Blood in the there's blood in the urinal hematuria, and is one of the most common sort of findings in Alports. There is anomalies in around ABP, in around arterial blood pressure. There is and can be swelling in the legs, ankle, feet, and in around the eyes. and the signs and symptoms may differ, but it is based on age, gender, and inherited type of Alports. In around as part of the Alports diagnosis, there is it is where analysis of signs, symptoms, and family history is important or can be important. Urine investigations can allow identification in around abnormalities in around biochemical parameters or biochemical constituents or components. Glomerular filtration rate or GFR, blood investigations to understand how well kidneys or renal are filtering toxins from the blood is and can be important. At the same time, a renal biopsy is and can be done where healthcare professionals will remove a tiny piece of the kidney, the tiny piece of the kidney with a special sort of a needle, and through cannulation there is an observation or under after cannulation there's observation through microscopy, an investigation of histopathology. In as part of vision investigations. Vision investigations can help to evaluate where vision can be affected, and in as part of in around genetic investigations. So genetic investigations, they tend to help confirm the diagnosis of confirm the diagnosis and of the specificity of Alports and the genetic and in around the genetic mutation. And as part of the diagnosis recommendations, so recipients with hematuria and lamellated glomerular basement membrane or hearing loss are likely to have Alports and should be offered genetic investigations for mutations in across three Alports genes. Patients with focal and segmental glomerulosclerosis should also be offered genetic investigations for mutations in Alports in addition to podocyte-related genes. All three gene mutations, COL4A5, COL4A3, and COL4A4 genes should be investigated in individuals undergoing genetic investigation for Alports. And genetic testing or investigations can be done through high throughput sequencing through laboratory investigations such as Sanger sequencing. And as part of renal biopsies, so podocyte hypertrophy and stiffness of the capillary wall are frequently observed, associated or not with presence of tubular red blood cell casts. Focal and segmental thickening of glomerular basement membrane with a progressive enlargement of the mesangial stalks is and can be important as part of biopsies. Clusters of interstitial foam cells are frequently found or identified in Alports recipients. Immunofluorescence can be important. It can be important as part of biopsy investigations. However, literature has formed that immunofluorescence is initially negative. As far as pharmacotherapeutics, There is no specific treatment for Alports. The goal is to manage the symptoms or symptomologies and help slow the progression of renal disease. ACE inhibitors or angiotensin receptor blocker pharmacotherapeutics are used to control ABP. Diuretics have been used in some in cases of Alports. Solid organ transplanted recipients with Alports is usually in Alports recipients is and has been successful, and is considered as one of the sort of more positive form. One of the better forms of KRT or RRT. Novel novel pharmacotherapeutics. Current standard of care for recipients with Alports is and has been with angiotensin blockade in these recipients with overt particular symptomologies. Treatment at earlier stage should be considered in males with X-linked Alports who have COL4A5 genotype associated with early progression to ESRD or ESKD, and with a family history. Initiation of angiotensin blockade while renal function is still normal. Delays of the onset of ESRD or ESKD. In transgenic Alports laboratory mice investigations, anti-microRNA 21 treatment had reduced glomerular inflammation and impaired renal fibrotic pathways. Angiotensin blockade is generally inexpensive and broadly available and effective through oral administration. In as part of the summary then of this and in around Alports. So there are still issues in the diagnosis and management of patients or recipients with Alports. Secondly, mutation database initiatives could be or can be helpful to help determine and explain how mutations are autosomal dominant or autosomal recessive. Thirdly, whole genome sequencing, as for example, in again Sanger sequencing, is and has been important and promising because it examines or investigates all three Alport mutations. Fourthly, laboratories should sought to improve diagnostics to ensure detection of mutations in amongst Alports. That draws this audio podcast to a close. This audio podcast as part of the conglomerate. This audio podcast is part of a Labostic Shystorm. This audio podcast is part of a Storm and Battle and Chronicle. This audio podcast is part of the Ztormish series or edition, and this audio podcast is part of a Storm Labostics production.